Hi everyone. This is part two of microbial control. And the last thing we talked about was how we use temperature to physically control the growth of microorganisms. And so we're going to continue on to the physical control topic. And here we're going to talk about osmotic pressure. So when we were talking about microbial growth and we talked about osmosis, this is the reason why we did that. And if we're trying to control the growth of microorganisms, hopefully you remember your osmotic conditions that are deadly to bacteria or microorganisms in general. So remember we said isotonic solutions and hypotonic solutions are survivable or desirable for bacteria, but when you get a hypertonic solution, that the cells shrivel up and the conditions can no longer be compatible with life. So here's just a description. This picture is completely from your textbook. And it's showing you here, if I put a bacterial cell in a hypertonic solution, that means that there's more solute outside of the cell than inside of the cell. And that also means that there's more water inside the cell than outside the cell. So osmosis says that water is going to move from high concentration to low. So the water in a hypertonic solution is going to move from inside the cell to outside the cell. And so that's going to be incompatible with life. And well before people understood anything about microorganisms, they used different kinds of osmotic conditions or hypertonic solutions to control the growth of microorganisms in food and to preserve food. So we would do things like dry out fruit, like you might have dried apricots or dried grapes and that turn into raisins. And just by simply putting the fruit in the sun or in a warm place, the water is pulled out of the cell and you just get drying. And so what's going to happen is the inside of the dried fruit is so hypertonic that microorganisms won't be able to grow. We also do things like we salt meats. And so putting salt, a high concentration of salt on the outside of the meat will draw the water out of the cells. And again, it preserves the food because it kills the microorganisms. We've done things like canning fruits or canning different things to be able to you put a lot of sugar in it. So what happens? You take the fruit and it pulls the water out of the fruit, and it's a way to preserve. In fact, we call that preserves. And that high amount of sugar in like jellies and things is a great antimicrobial because it creates this hypertonic solution. So osmotic pressure, high osmotic pressure, which means putting something in a hypertonic solution, is a great way to control microbial growth. And we continue to do that. Even things like peanut butter, you can put that at room temperature because it's so hypertonic. So if it wasn't hypertonic, bacteria would be able to grow in there a lot more easily. The next physical method of control I wanted to go over is the use of filters. And you can see here, again, these pictures are from your book, so you can go read about this in your textbook. But there's something called a HEPA filter. HEPA filter means high efficiency particulate filter. And HEPA filters are designed to filter microorganisms out of air. And here they're showing you different kinds of HEPA filters. And so here, you're familiar with HEPA filters because they're a lot of times on our heating and cooling systems in our houses, vacuum systems, vacuum cleaners a lot of times have HEPA filters associated with them. We talked a little bit in lecture about operating rooms. And so if you have an operating room, the air going into the operating room needs to be filtered, and that's going to be filtered through a HEPA filter. If you have a room where you have somebody in quarantine because they have an infectious respiratory disease, that air is going to be filtered as it goes out of the room. And so again, that would go through a HEPA filter. Here they're showing you there's some different kinds of HEPA filters, and you don't need to know the details here. Excuse me. But there's different kinds of HEPA filters that, sorry, glitch there. Yeah, so we're talking about HEPA filters, and that there's different kinds of HEPA filters, that some are designed to remove bacteria. Some are actually designed to remove bigger things like pollen. Some can trap bacteria, and there's some really specialized ones that can trap viruses. But you can imagine that something that can trap that small has to be probably not something we would use all the time. You know, it's probably pretty fancy. But HEPA filters are the kind of filters that filter air. The other kind of filter we use in the lab a lot of times are membrane filters, and membrane filters are designed to filter liquids. And so if I'm in the laboratory and I need a solution really fast that's sterile, I might not have time to run it through the autoclave. So you can pour it into the top of one of these containers that are sterilized. And here's a membrane filter, and this is a vacuum pump, and it's going to pull the liquid through to the bottom part here, and any bacteria in there will get trapped. If I want to do a really small volume, I can use a membrane filter on the end of a syringe and push that liquid through there, and you'll get sterile liquid coming out the end. And, when I worked in research labs, a lot of times we would use membrane filters if we had heat-sensitive liquids, or if I needed something really quick, or if I just needed a really small volume of something. So membrane filters are probably more common in research settings. Okay, so on to the next slide. Having a problem getting my thing to go forward. Yeah, so what I have here are I want to talk a little bit about surgical masks, because they are, a form of filter. And it says here there's two main types of masks, the face masks and the N95 respirators. And when I used to talk about this lecture a couple years ago, not as many people had firsthand experience with so many different face masks, and respirators than we do now since COVID happened. And one of the things that I think most people know from now, when we look at when a surgeon's wearing a surgical mask, what they're trying to do is they're trying to prevent reduce the blockage of droplets from them into the patient. We're not worried, the surgeon's not worried necessarily or usually about the patient infecting them, but you're worried about, you have an open body, and if you talk, we automatically make these tiny little droplets that can spread microbes all over the place. So these face masks, which I wrote here as surgical mask, but these surgical face masks are mostly designed to prevent the spread of the contamination from the person wearing it. So when we were talking about wearing face masks for COVID, if you're wearing this kind of face mask, it can protect you a little bit, because it can protect you from droplets of somebody who's close to you, but more likely it's going to protect other people from your spreading droplets. So if you're positive for COVID and you're trying to prevent the spread of droplets, wearing one of these face masks can be very, very helpful. The other ones that you've heard of are the N95 respirators. And these have a little bit more fitting that goes along. You want to fit them a little bit more tightly than these face masks. You might also hear some called KN95. And it's just that they're made in different countries, and some of them have a little bit different numbers. But the N95s are the ones made in the USA. I think a lot of them we find are KN95s made overseas. But what they're designed for is actually trapping microorganisms. So they fit more tightly and prevents the wearer from inhaling smaller airborne infectious particles. So this is really what you use to protect yourself. So if you are concerned about the environment you're in and you want to reduce your chances of getting colds or COVID or influenza, wearing an N95 is the kind of mask you should be wearing. And so these are what we would call a HEPA type filter. They're actually trapping microorganisms because they have small microscopic holes in them. That being said, even wearing a face mask like this, a surgical mask, can protect you to some degree against viruses, because one of the ways that we get viruses is just touching our face and touching our nose and transferring them to ourselves. And so wearing any kind of mask is going to reduce your chance of getting a disease, getting a respiratory disease. And it talks a little bit about things about how to handle masks. And I think people are getting really good at knowing how to do that because we've had a lot of practice with COVID. The next topic I want to talk about, the next method of physical control, is using radiation. And radiation, like I said, is a physical way to control microbial growth. And if you look at this scale of radiation, we have all different wavelengths going on, and between 400 and 750 nanometers, there's the wavelength of light is in the visible range. This is what we can see. And so you look at this scale right here, and that's where our visible light ranges are. Now, if we go to lower wavelengths, we get into something called UV, or ultraviolet light, and that's expanded out here. And this is what we would call non-ionizing radiation. I think I have it right here, first one. The non-ionizing radiation is what we would call UV light. So I typically would say non-ionizing radiation / UV light. Well, these lower wavelengths of light can actually damage DNA. And so when you hear about radiation being able to cause cancer, it's because the radiation damages DNA. And if you damage DNA in a gene that controls how fast cells divide, or controls the cell cycle in some way, then you actually can cause cancer. So this ultraviolet light does actually damage DNA. It kills cells, whether they're our own cells or bacterial cells. But because these wavelengths are still there, because of the particular wavelength of light, they actually don't penetrate objects very well. So when you think about it, ultraviolet light, like that seen in sunlight, it can cause cancer. But the kind of cancer it causes is skin cancer. And that's because it only affects the surface. It can't penetrate and give us things like leukemia or liver cancer or something like that. So ultraviolet light is actually very effective at microbial control. Look at that, even within the range of ultraviolet light, in these larger wavelengths is where sun is. And then you get into the lower parts of that is tanning, and then even lower is where you get the effective bactericidal wavelengths. And so typical sunlight won't be bactericidal, but when you get a little bit lower, it is. But all of these wavelengths can cause cancer, including that tanning zone, which is why we discourage the use of tanning beds and laying out in the sun to get tan. If we go to lower wavelengths, and just as far as microbial control, ultraviolet light, the situations I can think of it being used as, I might sterilize a tabletop. So earlier in this video, or earlier in this PowerPoint, we were looking at the biosafety levels, and somebody was using a biosafety hood here. Where we go? Right here, and here, where they put their bacteria and samples inside here. And then when they're done, they turn on an ultraviolet light and they expose this whole inside area to ultraviolet light, and it will kill any bacteria that are left on the surface. In BSL-4 situations, you might even have an entire room that has ultraviolet light exposed to it. Now, the people have to leave the room, but you can actually decontaminate the surfaces of the room by shining ultraviolet light in there. The other uses of UV light I can think of go with you might have a water purifying system in your house if you've had a problem with bacteria in your water. And let me get ahead of here. If you've had bacteria in your water, and you might have a UV unit on your water supply that the water runs through like a little UV light, like a little area that's exposed to UV light. Now you'd have to take the water and make it into a really thin stream of water because remember, it doesn't penetrate very well. I've seen a situation where apple cider was sterilized using UV light. And again, it's kind of like a coffee maker where you have a really thin stream of water or apple cider or whatever going through the UV light sterilizer. When we're looking at other forms of radiation, the X-rays and gamma rays are very effective at microbial control. And these are the lower wavelengths of light, and we call that ionizing radiation. Ionizing radiation works the same way. It damages DNA, but it's very effective at penetrating objects, particularly gamma rays. Although we know X-rays can penetrate objects because we can see inside the body. And both of these can be used to sterilize because they can penetrate objects and they can inactivate endospores. So radiation, let's see, ionizing radiation can be used to gamma irradiate food. And you might see in Wegmans, you can go in the freezer section and buy like a tube of hamburger, and the hamburger's been irradiated. So if you want a product that you want to keep around, has a little bit longer shelf life than regular hamburger, you might want to buy that. I know they've tried things like strawberries and different fruits to irradiate them, but the public isn't real keen on irradiated food. And I think, when we think of radiation and our food, people get really nervous. But irradiated food is not dangerous in and of itself. Think about if I took hamburger and I shined light on it, I could turn the light off and it doesn't have residual light in it. And it's the same thing with food. If you shine radiation on it, it can you can kill microbes, but the food itself is not radioactive. So irradiated food can be a useful thing. The other thing, the big thing that we use like ionizing radiation for are, when you look at those sleeves of Petri dishes that we get in the lab, somehow they got sterile. But if I put them in the autoclave, they would melt. So what they do is they put them in those bags, and then they put them in big boxes, and then they put them inside of a chamber that has ionizing radiation, that has gamma rays. And you can sterilize plastic things or paper things or whatever because the gamma rays will penetrate. So all of those Petri dishes and a lot of lab supplies are gamma irradiated. Now these are really powerful radioactive rays, and the radioactive sources are pretty dangerous. So this is something that would be done in only a professional situation. Non-ionizing radiation, UV light, is something that we could use at home as long as you knew how to use it safely. Let's see what's next. Oh, I just want to touch on microwaves because microwaves are actually in here. And I have had people talk about using microwaves to control microbial growth. And I've actually even heard it recommended that you put your sponge in the microwave because it will kill microorganisms. Well, it turns out the reason why it can kill microorganisms in your sponge is because microwaves heat up the sponge. But the microwaves themselves don't kill microorganisms. And so that was one of those things you saw on on Facebook and things like that. Put your sponge in the microwave. It'll great at killing microbes until too many people left their sponges in too long and they caught on fire. And so now people say, probably not a good idea to put your sponge in the microwave. So microwaves do not kill microorganisms, but they do generate heat, and heat is very effective at microbial control. The last thing I want to show you about radiation is one of my students a few years ago was a cleaner at Corning Hospital and was involved in the use of this Xenex robot. And these robots that cost, like $100,000 or somewhere around there are being used in hospitals more often because they make they generate ultraviolet light, non-ionizing radiation, and they can be used in rooms to kill microorganisms. And this is something that has been implemented due to so many antibiotic resistant organisms being in our environments. And these can kill microorganisms without treating them with chemicals or drugs. Now remember, when these are being used, the people have to be outside of the room, so they put these little warnings, don't go in the room, because UV light is really dangerous for us also. But this is something you're going to see routinely in hospitals if you haven't seen it already. There is a summary right here on physical control in your textbook, and there's also a matching that you can do in your textbook about physical control. Yeah, so we're going to talk about, actually, I'm going to start right here with surfactants for the next video. So we'll call that quits right now.